Hello everyone. Welcome to another session on biochemistry, and here we are going to deal with ribonucleic acid, the second group of nucleic acids. In the previous presentations we looked into deoxyribonucleic acid, the structure, the characters, etc. And this presentation onwards we will be looking into the ribonucleic acid, the different type of ribonucleic acids, the structure, the characteristic features, and the functions. As we can see, the ribonucleic acids, they are another set of what you call nucleic acids. It is a polymer of ribonucleotides which are held together. The ribonucleotides are held together by 3' 5' phosphodiester bonds, just like DNA. But what is the difference? Most of the RNAs, they are linear and they are branched. And okay, so ribonucleic acids, mostly they are linear, unbranched polyribonucleotide chains, and the ribonucleotides are being bonded together by the 3' 5' phosphodiester bonds. Okay, we can see that the RNA, some are branched, for example, lariats, the RNA forming lariats during intron processing, okay, processing of the RNA towards the final stage, okay, the post-transcriptional processing of RNA. During that time, the RNA forms a structure like a lariat, okay, there it seems to be branched. Some RNAs, they are circular in nature, for example, those in the viroids, right? Then so what are the differences between RNA and DNA? We have seen that the DNA and RNA both are polymers of nucleotides, one being polymer of deoxyribonucleotides, while the other being polymer of ribonucleotides. Chemically they have so much of similarity because internucleotide bonding, it is 3' 5' phosphodiester bond. But what are the differences? We can see that in the case of RNA, it is mostly they are single stranded. There are double stranded, there are exceptions, but mostly we can see they are single stranded as against double stranded DNA. Okay, in DNA also we can see single stranded DNAs, but again that is an exception. So usually in most of the cases we can see DNA, it is double stranded, while RNA, it is single stranded. Then another important feature is the type of the pentose sugar present in these nucleotides. In the case of DNA, the pentose sugar is deoxyribose, while in RNA, it is ribose sugar, which form the pentose sugar in the nucleic acid. Okay. Then regarding the nitrogenous bases present, DNA contains adenine, thymine, cytosine, and the guanine. Okay, the A, T, C, and G. But in the case of RNA, when it comes, it contains adenine, uracil, cytosine, and guanine. That is, instead of the thymine, the RNA has a uracil in them. Right. Another feature, it is the non-obeyance of the Chargaff's rule, or it doesn't follow the Chargaff's rule. Due to the single-stranded nature, we can see that the purine and pyrimidine ratio, it is never is equal to 1 in the case of RNA. Okay, in DNA, the like purine is to pyrimidine, the ratio, it is equal to 1. It is a characteristic feature of DNA and it is very specific to each species. But the RNAs, it never follows this particular rule. Okay, then sorry. Chemically, we can see that this DNA, it is more stable than RNA. RNA is metabolically active and they undergo translation process later when it comes to cytoplasm. Apart from that, we can see that the RNA, they are susceptible to alkaline hydrolysis. That is, the strong alkali, it can hydrolyze RNA to produce 2', 3' cyclic diesters. And it is due to the presence of a hydroxyl group at the 2' position of the ribose sugar. Okay, ribose sugar, it is having a 2' hydroxyl group in the case of RNA, while in the case of DNA, the deoxyribose is having only a hydrogen on the 2' position. Okay, because of this difference, what happens is the RNA, it is susceptible to the alkaline hydrolysis. Fine. Then we can see that when we quantitatively measure the DNA present in an organism, we can see that every cell of an organism will have the same DNA content. Okay, but when we quantitatively measure RNA, what happens is it may differ from one to the other cell of the same organism. Okay, so that is again a difference in the case of DNA and RNA. Okay, I hope these are clear. Now, when we speak about the classification, the RNAs, there are different types of RNAs based upon the physical properties, based upon the function they perform, right? So the ribonucleic acids, they can be divided into two functional groups based upon the physical properties and the functional aspects, and they are major RNAs and minor RNAs. Major RNAs, as you can see, they include viral RNA, mRNA, the mRNA in the sense messenger RNA, the ribosomal RNA or rRNA, the transfer RNA or tRNA. While minor RNAs include small nuclear RNA or snRNA, small nucleolar RNA or snORNA, small cytoplasmic RNA, telomerase, actually it is telomerase RNA, and the ribozymes. Among these, we can see that viral RNA, it is genomic in nature, that is, it is genetic in nature, it is hereditary. That is, in the case of viruses, the viral RNA, they have a very important genetic role. And apart from that, because of this genetic role, they can self-duplicate, they can actually replicate themselves. Okay, just like DNA undergoes replication, the viral RNA can also undergo the duplication. But RNAs in general, they don't have the ability to self-duplicate. Okay, they cannot make copies of themselves, unlike the DNA. But the viral RNA is an exception where it can take up self-duplication. It can make a copy of itself. Okay, just like DNA replication, the viral RNA can also take up replication. And since viral RNA has a very important genetic role, or it is what you call the main hereditary material in the case of viruses, they are known as genomic RNA, while the rest of the complete different types of RNAs are referred as non-genomic RNAs. Okay, now that is one unique nature of viral RNA. All others are non-genomic in nature. Among the non-genomic RNAs, we can see that the ribozymes, which is a minor RNA type, it is enzymatic in nature. Okay, they are the only ones which are enzymatic. That is, they like play, and they actually have a very important role as an enzyme. Okay, ribozymes. Fine. Now, coming to these, the mRNA, you can see that all these RNAs, like except for this viral RNA, all these RNAs, they are synthesized from a DNA template. Okay, the DNA, double-stranded DNA usually gets separated, the double strands get separated, and one of the strands, it forms the template strand, and based upon the base composition or the base sequence on the template strand, RNA is produced. Okay, all the non-genomic RNAs are produced, are synthesized by by the process of transcription from a DNA template. Okay, this transcription, it can be defined as a DNA dependent RNA synthesis process. Okay, it is a metabolic process. So this one is actually the this is how the RNAs are produced. Now, when you speak about mRNA, it is highly unstable. Metabolically, it is highly unstable. They are synthesized from the template strand on the DNA inside the nucleus, and these mRNA, it is then carried to the what you call cytoplasm, and it becomes a part of the protein synthesizing machinery. While in the case of tRNA and rRNA, they are produced again from the DNA itself by the process of transcription, but they are metabolically stable. And what happens is they do not code for proteins. Okay, these do not code for proteins, and they remain as the RNAs itself. While mRNA, it gets translated to proteins, and the life cycle, the lifespan is very fixed. Okay, so this is about general aspects about the ribonucleic acids and the different types. In the next presentation, we will see the different types of RNAs. Okay, thank you.